To view this video in Malayalam, you can click the link given above. Welcome to Biology My Passion. I am Soumya Harikrishna. We are now learning the chapter Cell, the Building Block of Life from the new NCERT textbook. Okay, spontaneous movement of molecules from a region of higher concentration to a region of lower concentration is called what? The diffusion. Osmosis is the movement of water molecules from a region of higher concentration to lower concentration through a semi-permeable membrane. Three conditions are there. Now, how will we say that the concentration is higher inside the cell or outside the cell? For that, here membrane means the plasma membrane. That's why we are considering the movement in the cell. Imagine we have three liquids here. Okay, A, B and C. What I am drawing as dots are the solutes. So this solvent is water. In that I have added solutes. Solute can be salt or sugar or any kind of molecules. We can call it as solute here. So in this it is very less, in this it is moderate, and in this it is very high. So you know that if you look at this compared to the water, suppose this level water I have taken in all the three beakers. In the first one, water is more, solute is less. Here solute is more, but still water is also there. Whereas in the third case, water is less because solutes are more. So whenever solutes are added more and more to a solution, the amount of water concentration is decreasing. So this has the least water concentration, this has the moderate water concentration, and this has the same water, more water concentration. Now imagine we are introducing a cell into this solution. So suppose a cell is added here. Okay, I am telling about an animal cell. This cell now has some amount of solute in it. Okay, so but that is more than the outside. That means here water is present, but more water is present outside, because outside condition is a dilute condition. So here we say the water will move inside the cell and the cell will swell up. Because outside more water than the cell, so water comes inside by osmosis and the cell swells up. When more water enters, naturally the cell swells up. And this process is called osmosis, or more specifically we can say endosmosis. Endo means inside. The osmosis happening towards the inside of the cell. This solution can be called as hypotonic. Tonicity is the concentration. Hypo means below. Less than the cell, the concentration. That means hypotonic solution has less solute, but it has more water in it. Water concentration is high. Whereas when it comes to the second case, we are introducing a cell, but outside the cell and inside the cell, water is almost the same. So we can call it as what? Isotonic solution. Iso means same concentration like that of the cell. Now what about the movement? The molecules may come inside and go out, but the net movement will be the same, because inside and outside it is the same concentration of water. Now coming to the third case. When we introduce a cell inside, we know that outside the solute is more, water is less, whereas inside more water. So as a result, the water will move out of the cell, because inside more water, outside less water, water will move out of the cell and cell will shrink. And we can call this solution as hypertonic solution. So you understood the concept of hypotonic solution with a higher concentration of water than cell, that's dilute we can say. So water will go from the outside to the inside cell, making the cell swell up. Second is isotonic, similar concentration of water with that of the cell, so there is no net movement. Third is hypertonic. As a result, there is movement of molecules of water from the cell to outside, because outside less water than inside, so that is called a hypertonic or highly concentrated solution. So in the textbook there is an activity given. You have to take potato of the same size, and we are putting it in different solutions. One is highly concentrated, or the solution to which we have added some solute already. Imagine you are taking some water in a beaker, you are putting 1 spoon of salt into it and stirring it. The other one, you are keeping the normal water without adding anything. To these two, we are introducing the similar size potatoes pieces, and we are waiting for a few hours. Before adding the potatoes, we can take the initial weight of the potato. Then after some time, we are seeing the weight. After a few hours, we take out the potato pieces and weigh them. We can see that where we kept in the normal water, the potato size has increased or the weight has increased because it has absorbed water inside because of the hypotonic condition outside. Whereas where we added solute or salt, the potato weight must have gone down because there outside is having less water than inside, so water moved out. So this is the reason behind this. The similar thing we experience in our day-to-day life. When you are soaking pulses, you know that whether it is chana or moong dal, anything that we put in water, green gram, we put in water for soaking. After a few hours, you can see it is swelling up. Or dry raisins, if you put it in water, after sometimes when we look, we can see they are swollen up. Why? Because they took in water because we are soaking them in normal water only. But at the same time, we are pickling it. In the salt water, if you are putting any of this, you can see that they are shrinking. Especially the tender mangoes. Once you are putting it in salt solution, they shrink because they lose water because outside it is hypertonic solution. In all these three situations, we are explaining about osmosis because we discussed the movement of water, and all these three cases water moved from higher concentration to lower concentration. Exception is this. And the third condition is all the time the water was moving through semi-permeable membrane or the cell membrane. So this is an example of osmosis. In the first case, water came in through osmosis, so it is called endosmosis. Endo means inside. Whereas in the last case, the water went out through plasma membrane or by osmosis, so it is called exosmosis. Let us now discuss the ultrastructure of the cell membrane, or what is it made of. We learned it is a universal feature of all the cells, and it's a very thin membrane protecting the contents of the cell from its environment and also allowing the movements of substances. So the thickness of the membrane is only 7 to 10 nanometer. Nanometer is a very small quantity. It is equivalent to 10 to the power of minus 6 meter. That means 1 million times of a meter. That is 10 lakhs times that of a meter. If you divide, that is 1 nanometer. So here it is 7 to 10 nanometer thickness is there. Now, what is it made of? It's made up of mainly two substances. Lipid is nothing but fat, or proteins also. So lipids and fat together form the plasma membrane. So we call it as a lipoproteinaceous membrane because it is made up of lipids and proteins. Apart from that, some amount of carbohydrate is also there. Now, one molecule of lipid, if you see, it has a structure like this. It has a long tail and a head part. This head part is water loving, hydrophilic. It loves water or it attaches with water. Whereas the tail is hydrophobic, water hating. So when the lipids are arranged, there are two layers of lipids, and these two layers are arranged in such a way that the water loving head is towards the outside and the water hating tail is away from water, because we know outside and water may be there. So it does not like water, so it is kept inside. So both the membranes are keeping their hydrophobic or water hating tail inwards and the head outside. Now I have drawn only these two layers of lipids only here. Now how are proteins arranged? Proteins are integrated in between this or tucked in between this, or there are proteins on the surface also on the lipid bilayer. Okay, so proteins can be either integrated inside or on the surface. On that there can be carbohydrates also. These proteins are allowing the movement of substances in and out. And the protein, when it is allowing substances to come, I told it is selectively allowing substances to come in and out. These molecules can move inside this lipid bilayer, like proteins can flip, taking a molecule from here, rotate and bring it here, or it can skew through the membrane. Anything is possible. That's why it is fluid in nature. Fluid means what? Anything that can flow. Here the molecules can flow. It's not rigid structure. So it is called a fluid mosaic model. Mosaic means the proteins are scattered like patches. You know, mosaic floor we see. There are a lot of chips there. Mosaic chips are there, right? So very patched appearance is called a mosaic. So here is a fluid nature and also mosaic nature. So this particular structure of the plasma membrane is referred to as fluid mosaic model. This gives a lot of flexibility to the cell membrane which allows certain functions also. And the proteins are the gatekeepers. They are the ones allowing the substances inside and outside. Next in your textbook given is an activity that you usually do in the practicals. One is a observation of animal cell or chick cell, the other one is a plant cell. For that we take either onion peel or we take rio leaves. So you can see that plant cells have an outer covering called a cell wall, whereas that is lacking in animal cells. Plant cells we usually use safranin as the stain, or sometimes iodine if we want to see the nucleus clearly, whereas in animal cells we use the stain methylene blue. In the coming video we will learn about the next part of the cell, that is cell wall. Hope you understood all the concepts I discuss here. Please help me reach this video to many people. Share with your friends, or you like, that will also give more reach. And if you have any doubts you can comment on my video, I will reply to you. If you haven't subscribed yet, please do subscribe. Thank you for watching Biology My Passion.